Okay. Could you tell us a little bit about where you're from, where you graduated undergrad, and your current research interests in the Young Lab?
Yes, for sure. I would like to start by saying thank you so much for doing this.
Thank you for coming.
It's a great pleasure to be here and be a part of this amazing series thing that you guys have organized. I was born and raised in India. I did my undergrad in biotechnology and masters in stem cells and regenerative medicine, both masters and undergrad from India. Manipal University, I'm not sure if you've heard of it, but really good university. And then I decided that I'm really interested in understanding the brain, which is very complex and beautiful. And so I moved to the US, did my PhD at University of Colorado, Denver. And then there I studied neurodevelopmental biology and stem cell biology. So specifically, my PhD was on understanding development of blood vessels that provides nutrition and oxygen to the brain. And so while pursuing my PhD, I got really interested in understanding how cells in the brain keep the brain healthy. And then when things go wrong, how do dysfunctional cells then contribute to brain disorders? So I studied neurodevelopment, but I got interested in adult brain functioning and adult brain disorders, and that interest brought me to the Young Lab. And then the Young Lab, just to give you an introduction, specializes in human-induced pluripotent stem cells and CRISPR-Cas9 genome editing. They use that as tools to address questions associated with Alzheimer's disease. So I was really interested in studying Alzheimer's disease, and so I joined the lab as a postdoctoral fellow. And then I studied, I used these tools that I just talked about. And to just give you an overview of Alzheimer's disease, it's of course very complex, but very simply put, what happens is, think about a garbage disposal system. So cells in the brain also function in a similar manner. They have cleanup and recycling systems in place. And when that goes wrong, there is buildup of waste materials outside the cells and also inside the cells. And that happens over a period of number of years. So you can imagine that leads to like really stressed and damaged cells. And so ultimately what happens is people with Alzheimer's disease, like patients, start losing their ability to think, memory, and you probably already know this, you may have heard about this, just daily functioning capability, because cells are unhealthy, damaged, and they start dying, right? And so my interest in the Young Lab, again, very simply put, is understanding this cleanup and recycling system in specific brain cell types. And how can we understand the system? How can we correct it? To essentially, ultimately, our ultimate goal is to find an effective disease-modifying therapy for Alzheimer's, because there are none. There are drugs out there, definitely, which are promising, and I should probably say that loud, and they alleviate symptoms, so that's great. And there are drugs that are coming up that decrease cognitive decline, which is again great, but we're still not there yet. None of them significantly reduce progression of the disorder, which it's such a debilitating disease that people suffer for many years. And so what we want to do as researchers in the field is step in and make their lives a little better. But you have to understand the brain is really complex, and so understanding neurodegenerative disorders is as complex. But we are getting there. And so just to end, answering this question, that was very simply put, right? It's very complicated, but the science term for that system that I talked about, cleanup and recycling system, is endolysosomal network. So that's my interest in the lab, understanding how dysfunction of that network in different brain cell types contributes to Alzheimer's disease. Yeah.
Thank you.
Yeah.
Can you introduce us to the SORL1 gene and how it affects cells?
Yes, absolutely. So the SORL1 gene encodes the protein sorola. And so it's a sorting receptor. And what that means is, think about traffic and traffic jams, right? So what the sorting receptor does is it prevents traffic jams inside cells. And it literally sorts, meaning all the biomolecules that enter the cells, it makes sure that these biomolecules reach their right locations inside the cell. So within the cell, you probably already know this, there are like subcellular organelles, right? And so there are many, and I won't go into details, but whenever something enters the cell, they need to like traffic between these organelles, and that's required for proper functioning of the cells. So SOROL1 here is one of the many sorting receptors, and what sorting receptors do is they make sure that these biomolecules reach, the correct destination essentially. And so how does that affect cells? So specifically in the brain, it's expressed by many brain cell types. So it makes sure that the cells take in whatever is supposed to be taken in from the environment. And that process is called uptake, so it can participate in that process, and then it participates in moving biomolecules inside the cell, again pushing them to the right location, and then ensuring what needs to be degraded goes into the organelle that degrades cells, and then these things then get cleared from the cell. So you can think about it as a traffic jam regulator. Yes. And then what does it mean? So what's the relevance to Alzheimer's disease, right? So SOROL1 is a very well-known and strong AD risk gene, and what that means is this gene has been identified as a gene that increases the risk of developing Alzheimer's disease. So it is strongly associated with Alzheimer's, that's why we study it. And also it has been shown through several decades that reduced protein expression has been seen in Alzheimer's disease brains of SOROLA. So that means that reduced SOROLA protein expression is associated with the disorder. And so we have clear evidence to show that a problem with SOROL1 gene is associated with the disease, but we're only starting to understand in details the function of this protein. And so our work and other researchers' work have shown that it has very important function, again in that system that I described in the beginning, the recycling and cleanup system in different brain cell types. So it has fundamental, very, very crucial roles and very different roles in different brain cell types, and it ensures that these cells are healthy and functional. And so one of our main goals in the lab, and one part of my research work, is to understand when you have reduced SOROLA or dysfunctional protein, how does that affect functioning of the cells, and how is that correlated with Alzheimer's disease?
What exactly is microglia, and how is it affected by Alzheimer's disease?
So microglia is actually one of it's a very important brain cell type, and you can think about it as very important part of the defense system of the brain. So it performs several functions. So if I were to define it, you can say tissue resident immune cells of the brain. So they're immune cells, specialized immune cells of the brain, and they live within the brain. They don't just sleep the brain and move around. And so these cells, let's talk about a healthy brain. So in a healthy brain, what happens is these cells are constantly surveilling the environment. And so like I explained before, you need to make sure that anything that's not required to be there outside the cells needs to be taken up by the cells and degraded and cleared, right? So microglia do that. And in addition to that, if there are any so-called danger signals, or in case of disease, microglia are the ones that get activated. And what that means is they change their state to a form where they become really protective, against, very simply put. And so they are able to, like I said, sense these danger signals and then enhance certain functions that enable them to clear these toxic molecules that start accumulating in the environment. And then in case of Alzheimer's disease, what happens is there are certain toxic proteins that keep getting accumulated over a period of multiple years. So what happens with microglia in a disease setting like this is they switch from being protective to actually being harmful to the brain. So they either get like overactivated or just overworked and overwhelmed, right? and then just start contributing to the progression of the disease. And so that's not good, obviously. And so what we're trying to do, and one of my main sort of research expertise and interest is in understanding how these cells make that switch in Alzheimer's disease and why that happens. And so, studying this cell type is so crucial because they are at the center of this, cleanup function. And so they clean up waste, and the main problem in Alzheimer's disease is waste buildup. And so remember, there are other cells that also clean up waste, but microglia are the primary cell type. And so there's a lot of interest in understanding these cells because people are trying to, engineer drugs and think about how can we utilize or leverage the ability of microglia to clear things from the brain, because that will possibly help in improving certain brain functions and at least let people live with some dignity. Yeah.
Can you briefly explain to us how you use gene editing like CRISPR-Cas9 to study SORL1?
Yes, absolutely. So again, let me explain CRISPR-Cas9 gene editing in very simple terms. It would be, editing the DNA, making changes in your DNA, right? And so how do you decide what you're going to edit? The decision is made. There are several factors involved. So in this case, we were interested in the SORL1 gene. We know there is reduced protein in an AD brain. So what you want to do is, as a researcher, you're thinking, well, can I recapitulate that in a dish? And so how do I do that using CRISPR-Cas9? So what you can do is you can make edits. Think about scissors, right? So you can actually cut the DNA at a place that ensures that you do not make the functional protein. So you could either do that, or you could do what is called as CRISPR-I. I won't go into details, but you can even repress the gene just to get reduced amounts of genes. So you can get no protein at all, and you can get reduced levels of protein. For our paper, what we did is we completely knocked out the protein. That's why in the paper you will see there's a lot of SORL1 knockout microglia. That word gets used a lot, right? Because we made SORL1 knockout microglia. And so again, I can tell you how we made it in one line. So what happens, what we did was we modified the DNA in a way where we cut the DNA gene at in a very early part of the gene. And so what you're doing is you're ensuring that the gene stops getting instructions to make the protein from a very early time point, and that ensures that you do not get a functional protein. So the idea is to get iPSCs, which is what we used, human-induced pluripotent stem cells, that have functional SOROLA protein, and they're called wild type. And then you have the SOROL1 knockout iPSCs that do not have the protein. So that's how you generally use CRISPR-Cas9, and that's how we generated our SOROL1 knockout iPSCs. So you start with iPSCs, which are your stem cells, and then you differentiate them. And the word differentiate essentially means you are programming them to become different brain cell types. And these protocols to program them are developed by researchers, or we develop some of them in our lab. And the idea is you take concepts from developmental biology, which is what I did my PhD in. And so from developmental biology textbooks and general knowledge of developmental biology, that there are certain factors that come from the environment, from different brain cell types, when a stem cell makes a certain cell type, right? So you add those factors when you're making these cells in a dish, and that's how you change a stem cell to a microglia or a neuron or different brain cell types. And so for this paper, the way CRISPR-Cas9 was used was we made iPSCs that have functional SOROLA, do not have SOROLA, so wild type and knockout iPSCs. Then you make microglia from them in a way that I just described, and then you use these microglia and then do your experiments. Does that make sense?
Yes, thank you.
Now let's dive into using what we've learned so far as to actually read the paper.
One of the most important components of reading papers is being able to understand figures, so let's explore that. Figure 6 here demonstrates the novel finding of this paper, that sorrel 1 regulates lysosomal function in microglia. So what's really being shown here? In subfigure 6a, we see what happens to surface LAMP1 localization. As seen in this figure title, with and without the compound calcimycin. So we can see that right here. LAMP1 is a protein that is basically used as a marker for lysosomal activity. We'll see it expressed with lysosomal membranes. Calcimycin is a calcium-based compound that triggers lysosomal exocytosis, a form of lysosomal activity where the lysosomes work to move waste out of the cell. Basically, we use calcimycin to induce lysosomal activity that we visualize with the fluorescent expression of LAMP1. If you're curious what CX3CR1 and DAPI mean over here, good catch. These are also protein markers. DAPI is used to visualize cell membranes, while CX3CR1 is a marker for immune cells, like the microglia that they're studying in this paper. Basically, DAPI allows us to see about where the cell is, and CX3CR1 is used to identify if that cell is an immune cell, microglia in this case, or not. In the first two columns of each trio of images, we're visualizing DAPI and LAMP1 together, and DAPI and CX3CR1 together respectively. As you can see, we have a little bit of pink here, and we have blue here, and here we have blue here, and green here. So that corresponds to green is CX3CR1, pink is LAMP1, and then the blue is DAPI. In the last column, we see all three markers visualized at once. This can look a little bit weird just because what they do is they overlap the colors, so sometimes you get a little bit of white where it should be the combination of of green and pink together. So it's a little bit confusing, but we got it. Let's move on to looking at these rows. For the top two, we have WT, which in this case means wild type, and pretty much any case in reading a scientific paper, it means wild type. HMGLS, which means human microglias, because we're studying here basically what a human microglia looks like in the wild. On the bottom two rows, we have sorrel 1, 1KO, HMGLS. So we have sorrel 1, which is a gene, 1KO, which is single knockout. So they're saying that these are human microglia cells with only the sorrel 1 gene knocked out using the CRISPR-Cas9 technology we discussed earlier. The inset rows, right here, they're basically just zoomed in versions of the cells that we see with a dotted line square around them. So it's saying like we're zooming in on these. You get a closer look. Now, what's interesting about this figure? Why would a researcher think to put this in their paper? Well, this subfigure visually shows the differences between wild type and sorrel 1, 1KO. You can see there's not much difference between the wild type and knockout cells in the without calcimycin category. You can see you still don't really see any LAMP1 being expressed, which makes sense because LAMP1 is, again, a way for us to see lysosome activity. However, with the calcimycin, there's a pretty big difference. You can see that in these wild type cells, we get a lot more pink going on than we do in these sorrel 1, 1KO knockout cells. This means that the wild type cells tend to have a lot more LAMP1 expression than the KO cells, meaning that they tend to have more lysosomal exocytosis than the cells lacking sorrel 1. But how do we know that this behavior is generalized across at least the majority of their data? Well, that's why the authors have included more than just a sample of visual results. Subfigure 6b here shows us the quantitative results of this study with surface LAMP1 localization with and without calcimycin. The reason why we see DMSO being used here, if you're a little bit confused, is because scientists typically use DMSO to dissolve calcimycin. It's kind of like the placebo, it's the base case, one where no treatment is being used. It represents the background noise of DMSO when working with calcimycin. So you can think of it as this is the blank, and then this is the blank plus a treatment. In this figure, they're measuring the mean fluorescence of LAMP1, which is basically on average how bright is a LAMP1 signal. We can see that without calcimycin, which is right over here, this section, there's not too big of a difference between the wild type and knockout expressions. We can see that if you look really closely, some of these points are kind of at the same level. However, once they use calcimycin instead of DMSO, we see a whole different story over here. The wild-type cells express much more LAMP1 than the SORL1 KO knockout cells do. This means that when they knock out SORL1, there's less lysosomal exocytosis occurring because we have less LAMP1 activity going on when we induce lysosomal exocytosis. You may be asking now, though, how do we know that this isn't just that knocking out SORL1 leads to less LAMP1 expression in general? How do we know that you can still express LAMP1 when you knock out SORL1? Well, that's where subfigure C here comes in. Here we see a Western blot, which is a type of study used to determine how much of a certain protein is being expressed in a more qualitative manner. You can kind of see how thick the line is and if it occurs at all. If you don't have a line at all, then that means that you don't have really any expression of that protein. They're using beta-actin here as a sort of control, just like the DMSO from subfigure B, just so you have a reference point. We can see that with LAMP1, both clones of the wild type and the SORL1 knockout are still able to express the protein. They're still able to express LAMP1. We all have lines of this. The knockouts just really aren't doing so in comparison to the wild type when calcimicin or lysosomal exocytosis is induced. To quantify their data, so we know they aren't pulling a fast one on us, they also include a small graph here showing LAMP1 expression normalized to beta-actin, so we can see that wild type and KO have about the same levels of expression. They're about the same here. Subfigures D through F further solidify the author's point. They show how certain enzymes associated with lysosomal exocytosis, specifically HEXB, CTSB, otherwise known as cathepsin B, and cathepsin D, CTSD, have decreased activity in SORL1 knockout versus wild type with calcimicin. Subfigures G through L show how interleukin 6, or IL-6, a protein that regulates immune function, levels change between different conditions. Subfigures G and J here show how IL-6 levels change with calcimicin treatment, while subfigures H, I, K, and L over here show how IL-6 levels change in response to inflammation from lipopolysaccharide, which is this LPS over here, and interferon gamma, which is the IFN gamma over here, stimuli. You'll also see that G through I is labeled as media, while J through L is labeled as lysate. In this study, they used a test called an ELISA, an enzyme-linked immunosorbent assay, which determines whether or not a specific protein is present. In this case, interleukin 6. In testing the media, they're testing what the cells grew in and what extracellular IL-6 was found, what happened to IL-6 outside of the cell. In testing the lysate, which is where they break open or lyse the actual cells, they're testing the presence of intracellular IL-6, or IL-6 that was present inside of the cell. We can see that extracellularly, or with the media, IL-6 levels differed drastically upon addition of calcimicin. I mean, here for DMSO, it's barely even there, but with calcimicin, we have a big jump. However, intracellularly, there's not much difference between addition of calcimicin or DMSO, our background level. Overall, we can see from subfigures G and J that there is at least some difference between SORL1 knockout and wild-type cell levels of IL-6, with extracellularly there being less in KO cells with calcimicin added, and intracellularly there being more in KO cells with either DMSO or calcimicin. Figures H, I, K, and L are a little bit more straightforward, with there being the same results intra and extracellularly. SORL1 knockout cells tend to have lower IL-6 levels in response to inflammatory stimuli. As you can see here, this bar is a lot lower than the other one. That's it for figure 6, but how was I able to get all of this information out of just a couple pictures and graphs? One thing that's super helpful in interpreting figures like this is just checking out the caption. It can be a little bit intimidating, there's a lot of text there, but if there's anything ever still unclear to you when reading papers, feel free to just go ahead and look it up. Sometimes the greatest barrier to entry for understanding papers is just not knowing the vocabulary presented. The easiest way to overcome this barrier is just by asking for help. Now let's take a look at their working model. A working model acts as a visual overview of what this paper is presenting. So this is the authors telling you that these are the mechanisms and functions and how we're picturing them. Here we can see a comparison between wild-type and SORL1 knockout human microglia-like cells. Sorla, the protein that SORL1 produces, over here, is represented by these orange circles, which you can see in the wild-type cells, but you can't see in the SORL1 knockout cells. Right off the bat, there are a lot of noticeable differences. For example, we have a lot of phagocytic receptors in the SORL1 knockout one, P2Y6R and TREM2. We have less trafficking from early endosomes to the lysosome. We have less lysosomal exocytosis in our SORL1 knockout cells, and there's a lot of differences that we'll get into in a second. With these SORL1 knockout cells having more phagocytosis receptors, this leads to greater phagocytic uptake, because as the receptors are being activated, the cell believes that it has the capacity to process all of this waste and says, yeah, we'll take those synaptosomes and amyloid beta proteins. However, loss of SORL1 has led to impaired trafficking of SIMPR, which you can see are these things right here. You can see that they're kind of struggling over here. And we also have less of them in the lysosome in general compared to this wild type. According to the caption, this is because we have less lysosomal enzymes coming from the TGN over here, which is the trans Golgi network, seen with these light and dark circles in the lysosome. You can see over here we have 4 of each. Over here we only have 2. So that really sucks for our lysosomes. Putting these two facts together, we can see that the knockout cell lysosomes are really struggling to manage this waste. There's a lot of it coming in and there's not a lot of it going out. There's fewer functional premature and mature lysosomal enzymes and more waste for the lysosome to traffic. As you can see, these ones are definitely faring much better than these ones. This leads to an overall decrease in lysosomal degradation and exocytosis, leading to waste accumulation in the lysosome. And you might be wondering, what does waste accumulation in the lysosome cause? Well, that's the finding of this paper. These wastes, specifically the amyloid beta proteins, those accumulations are associated with Alzheimer's disease, and they're known as amyloid plaques. So overall, this model shows that the loss of SORL1 really stresses out our microglia's lysosomes, leading to issues with cell waste degradation and, on a larger scale, neurodegenerative diseases like Alzheimer's. What do you think your results could mean for future research, or how we understand Alzheimer's effects on the body?
That's a great question. So, what I think is our research has not only identified a novel function for a known AD gene in a cell type that had not been, studied before in the context of SORL1. So we identified that loss of SORL1 actually causes lysosome dysfunction, but this also has broader implications for the disease. So we have shown that it's probably important now to focus on the lysosome and lysosomal pathways. So our study, I would say, informs future translational studies or therapeutic strategies that should, I think, focus on clearance pathways and how microglia can clear intracellular waste and probably utilize one of these pathways, specifically lysosomal exocytosis, can we modify it to make microglia more efficient in cleaning up waste in an Alzheimer's disease brain. Yeah, that's what I would say.
Do you have any upcoming plans for where you're going to take this research or your research in the future?
Yes, of course, always. So I'm actually working on my upcoming plans of the past 2 years through 2 funded grants and essentially taking the concept of lysosomal exocytosis forward. So like I said, my plan was to see, well, does this happen in AD patients? And so what I'm doing right now is I have access to iPSCs from Alzheimer's disease patients who were at different stages of the disease. And so we have access to post-mortem brain tissue from these patients and iPSCs from them. So then the idea is to look at post-mortem brain tissue. Well, are the lysosomes dysfunctional? You actually can do what is called immunohistochemistry, meaning in simple terms, you're labeling the microglia and then labeling the lysosomes and then doing certain experiments to see if there is a problem with these pathways. And then remember, we get iPSCs from the same patients. And so when you get iPSCs, you can actually, like I've described before, make microglia from them, right? And then run all these assays. The end goal will be to see if we see altered lysosomal exocytosis and are there ways to rectify it? And then I would like to highlight here, we're not just focusing on SOROL1, but we have lines or AD patients that have been ranked based on, they've been given a score, and it's called a polygenic risk score. So I can simplify that. So that just means, so I talked about SOROL1 as one AD risk gene, right? Now there are several AD risk genes. And so what was important, again, just think logically, right? So many AD risk genes. What you want to know is what is the combined effect of all the genes, because that's what happens in a human. And so that's exactly what we're trying to do. We're trying to understand the combined effect of all these variants on lysosome function. So these are AD patients that have been given either a high EPRS score or low EPRS score, and that just means we will be able to understand the combined effect of these variants on these specific pathways in a specific brain cell type. And then again, the end goal is to identify, to actually come up with tractable interventions. Like, can we modify this? Can this be tracked in like a live human AD patient? And so, and to do so, there's a long way to go. But the end goal is to, kind of say, hey, if we add, it's not as simple as you add one drug and you enhance this pathway and everything is lovely. It's essentially understanding what this pathway does and how can we, again, get that balance. But the main question right now is, do we actually see this phenotype? Because in our paper, it was an artificially created system, right? And it did tell us that reduced SOROLA is a problem. But then in AD patient cells, it's a more complicated system. And so you would want to know for sure that this process is altered, because if it's not, then you look at another pathway that is. But yeah, that's the plan, and that's where we are at. Yeah.
Thank you so much for chatting with us
No
today.
problem. Thank you.
Yeah.
It was lovely chatting with the both of you.